Hello, good afternoon, everyone. Okay, so let's start. We all have heard about the Nipah virus a lot. This has become an emerging kind of virus today and has caused lots of problems. So let's start. What does the Nipah virus mean? Let's come with the introduction at the first. Okay, this human Nipah virus NIV infection is an emerging zoonotic disease which causes several diseases in both humans and animals. Today, Nepal is also under the threat of Nipah virus, and obviously we must all get aware of it. Okay, so this human Nipah virus is an emerging zoonotic disease that has been transferred from the animals in both human and can be intertransferred as well. So we have to be very much careful regarding this Nipah virus. Nipah is a member of family Paramyxo. Okay, so Paramyxo, member of Paramyxo virus. So yeah, it was first recognized in Malaysia and Singapore during an outbreak from September 1998 through May 1999. It was first seen in Malaysia. People recognized and came to know about the Nipah virus first in Malaysia and in Singapore during the 1998 and 1999. Its name has been originated from Kampung Sungai Nipah, a village in the Malaysian peninsula where pig farmers became ill with the encephalitis. Regarding the structure, we can see here, this one is the structure of Nipah virus. Okay, this top type, like the yellow colored, okay, arrowhead is the fusion protein. This one is the glycoprotein. Okay, this one is the matrix protein. This one is the envelope. This one is the nucleocapsid. This one is the phosphoprotein, and this complete part is called as the virus ribonucleoprotein. Okay, yeah. So this one is the structure which is showing the okay genomic as well as the outer structure of the Nipah virus. Nipah has become a public health problem today. Yes, it has become a public health problem. Now, though Nipah virus has caused only a few outbreaks, it has affected a wide range of animals and has caused several diseases in and death in the people. Yes, even in the Asian countries, this Nipah has caused a threat. So we Nepalese should also be aware of this. Okay, we can see here the different kinds of outbreaks of Nipah virus in different years. It was first reported in the Siliguri during 2001, in Faridpur in 2004, in Manikchand and Rajbari in India in 2008, okay, and so on. Now, so we can see here the different outbreaks that has occurred, okay, because of the Nipah virus in several years. Coming to the morbidity and mortality rate due to Nipah, from 2001 to 2018, we can see uh, these rates. Okay, the recent outbreak has been seen in Kerala and has been thought that the has been caused by the dead bats, okay, from Chomero. Yes, the infection reportedly spread among family members and was passed into the others who has been in contact with the family. Now we can see here how does the transmission of Nipah virus occur. Okay, first of all, this theropid fruit bats are the natural reservoir. Okay, it is then taken. This is the theropid fruits. Okay, bats. The bats which eat the theropid kinds of fruits. Okay, these bats are the natural reservoir of the Nipah virus that get transferred into the pig, and from the pig it gets transferred into the human. Regarding the pathogenesis, the pathologic findings in the brain of Nipah encephalitis cases showed evidence of necrotizing vasculitis. It affects the brain also, causing the meningitis. Okay, and the main pathology appeared to be widespread ischemia and infraction caused by vasculitis-induced thrombosis. Alveolar hemorrhage can also be seen. Pulmonary edema and aspiration pneumonia were also encountered in the lungs. These may lead to pneumonia and acute respiratory syndrome, distress syndrome. Okay, it has similar kind of pathogenesis that of the coronavirus, but the new thing here that we can see is it causes the meningitis and encephalitis as well. This may lead to pneumonia as well and can cause ARDS, that is acute respiratory distress syndrome. Some of the common signs and symptoms that we can see after the Nipah virus attacks into the human, there can be the acute onset of cough. There can be the cough with shortness of breath. Somehow the similar symptoms that is seen in the asthma. There can be the severe headache as well, and some mental status can be altered as well. The high grade fever can be seen, diarrhea can be seen, muscle pain can be seen. Okay. For the diagnosis, treatment, and preventive measures, for the collection, transportation, and storage of sample, okay, the sample should be collected as early as possible, preferably within 4 to 5 days on onset of illness. The samples may be of any kind, throat swab that can be collected in VTM, that is called viral transport medium, and the urine approximately of 10 ml in universal sterile container can be taken. Okay, blood in plain vial, at least 5 ml can be taken as the sample, or CSF, that is also called as cerebrospinal fluid, can be taken at least 1 ml in sterile container. Samples can be safely packed in the triple container packaging and can be transported under cold chain in between the temperature of maintaining the temperature of 2 to 8 degrees Celsius to the testing laboratory with prior intimation. Before dispatching the sample, disinfection should be done onto the outer surface of the container with 1:100 dilution of bleach or with 5% lysol solution. These are the different kinds of diagnosis techniques which can be used for the diagnosis of Nipah virus. They are by the serological method, by RT-PCR method, by ELISA methods, by virus isolation, by neutralization test, and by immunohistochemistry. Yet no specific treatment of Nipah virus has been seen. Okay, so that is, the prevention can be done. We can maintain the preventions, and we can also maintain a mechanical ventilation, and different nursing techniques can be also helpful to prevent the okay, infection from the Nipah virus. These are some of the antiviral treatments like ribavirin, chloroquine, neutralizing antibody. These are some of the species and their efficacy can be seen here. Other thing can be, the further thing that can be done is the vaccine development activity, which should be done, okay? And these are some of the preventive measures: hand hygiene, infection safety, facial protection, okay? And practice of respiratory hygiene, gloves and gown wearing, okay? Respiratory hygiene, cleaning of the environment, and the intake of fruits bitten by animals should be avoided, and proper handling of the specimens should also be done, okay? Thank you.